Previously on Grasses and Classes. Brackish water is a solution made from the mixing of salt water and fresh water. Our Spartina alterniflora likes to grow in brackish water. We learned that salinity, or the amount of salt in water, can be measured in parts per thousand using a refractometer. Other substances in water, like chlorine in drinking water, can be measured in parts per million or parts per billion. We can weaken the concentration of a solution by diluting it with water. We also learned that a small change in concentration has a big impact on our waterways and the organisms that live in them. But how exactly can these small changes impact organisms? Is there a way for an organism to change so it can continue surviving in a changing environment? Tune in to the third lesson of Grasses and Classes to find out. Welcome to Grasses and Classes, brought to you by the Choctawhatchee Basin Alliance and AmeriCorps Northwest Florida Environmental Stewards. This is Lesson 3, Adaptations to Environmental Factors. Welcome back, explorers! In our last lesson, we learned how small changes in concentrations can have a big effect on our waterways. We also learned how CBA measures salinity, dissolved oxygen, and sediment in the Choctawhatchee Bay, which is also known as an estuary. An estuary is where the river meets the sea. Plants and animals that live in estuaries need both salt and fresh water to survive. Too much of either can harm them. We get our fresh water from the Choctawhatchee River and lots of creeks. Rain also adds fresh water. It mixes with salt water entering through the Destin Pass to create a brackish water solution. Can you remember other examples of solutions? What does it mean to dilute a solution? Pause the video now to discuss. Solutions occur when two ingredients are mixed together and are evenly distributed. Examples can include Kool-Aid, tea, and air. When we dilute a solution, we are making it weaker or thinner. What about when the salinity levels change in the bay? Can we dilute the entire bay? No, the bay is way too big, and weather plays a huge part. Sometimes plants and animals need to adapt to new conditions. Let's go check out a local habitat to see adaptations in action. An adaptation is the way a plant or animal changes in order to survive. Here we have Spartina alterniflora. We already know it is a native plant and it's naturally found along the shoreline of the Choctawhatchee Bay. Smooth cordgrass has many adaptations that help it survive in this environment. For example, when smooth cordgrass takes in brackish water, it can get rid of the salt through its leaf blades, leaving it with usable fresh water. The Choctawhatchee Bay has a range of salinity levels. Understanding what we have learned, where in the bay would we have the highest salinity levels? Where would we have the lowest? Pause the video now to discuss. The highest salinity levels would be on the west side of the bay where the Destin Pass connects to the Gulf of Mexico to the bay. The lowest salinity levels would be on the east side where the Choctawhatchee River empties into the bay. Do you think all these different areas have the exact same plants and animals? No. Where they are in the bay depends on the salinity levels. Plants and animals in our bay must be able to adapt to changing salinity. What are some other adaptations you can think of? Pause the video now to discuss. Camouflage is one common adaptation. This is when an animal or plant blends in with its surroundings. Can you find the animals above? Pause the video now to discuss. Another plant that has adaptations to live in a certain kind of water is Elodea canadensis, also known as pondweed. What kind of water do you think it lives in? Pause the video now to discuss. Freshwater. In order to see if it can adapt to a different salinity, we are going to look at its cells under a microscope. But what is a cell? A cell is the smallest unit of a living organism. Plants and animals are made up of millions of cells. Let's look at a few key parts of a plant cell. Open your folders to page 13, where you will find a diagram you can follow along with. As we go over the cell parts, it might be kind of hard to think about because they're so small. So we're going to compare the cell parts to things in your classroom. So take a good look around you. This is a cell wall. It surrounds the cell, protecting it from the outside and making it stiff and strong. What part of your classroom might this be? Pause the video now to discuss. That's right, the cell wall would be like the classroom wall. This is the cell membrane. It helps hold together and protect the cell, as well as controlling what can go in and out of the cell. What part of your classroom might this be? Pause the video now to discuss. The cell membrane would be the doors and the windows of your classroom. This is the cytoplasm. It is a watery, gel-like material which helps the various parts of the cell move around. What part of your classroom might this be? Pause the video now to discuss. The cytoplasm would be the air in your classroom. These are chloroplasts. They contain chlorophyll, which is a green pigment that captures sunlight. The chloroplasts use energy from captured sunlight to produce food for the cell. What part of your classroom might this be? Pause the video now to discuss. The chloroplasts would be your lunch boxes. This is a vacuole. It stores food, water, and various chemicals for the cell. What part of your classroom might this be? Pause the video now to discuss. The vacuoles would be storage areas like cabinets, cubbies, and desks. This is the nucleus. This acts as the command center of the cell, regulating and controlling all activities in the cell. What part of your classroom might this be? Pause the video now to discuss. The nucleus would be your teacher. Now that we know the different parts of a cell, let's learn about the different parts of the tool that we will use to see them today, the microscope. First, let's look at the eyepiece. The eyepiece is where you look through to see the enlarged object. Then, underneath the lens, we have the stage. This is where you put the slide with your object. Here are the stage clips to keep your slide in place. The stage is also an opening that allows light to pass through. Under the stage, a light illuminates your slide. The diaphragm between the light and the stage controls how much light comes through. Next, we have the objective lenses. These lenses bend light to magnify the object you're looking at. The microscopes we'll be using have 3 different magnification strengths. This is the base of the microscope. It supports everything and contains all the electronics. Here the arm supports the lenses and stage. When you first look through the eyepiece, your slide might look blurry. You can use the adjustment knobs to fix this. First, you can use the coarse adjustment knob. It raises or lowers the height of the stage, and it's good for finding the part of the slide you want to look at. Then you can use the fine adjustment knob to make small focus adjustments. Finally, here's how to safely carry a microscope. Put one hand under the base and hold the arm with your other hand. Now that we know more about our microscope, we're going to use it to look at Elodea canadensis. We're going to compare leaves that have been in fresh water versus salt water. But first, there's something we must do. We need to create a problem statement. A problem statement is a question that our experiment is trying to answer. So what do you think our problem statement should be? Open your student folders and pause the video now to discuss. Our problem statement today is, can Elodea adapt to a saltwater environment? Now that we know our problem statement, we can come up with a hypothesis. A hypothesis is an educated guess about what will happen in an experiment. We can express that by using an if-then statement. Can you come up with a hypothesis? Pause the video now to discuss. Your hypothesis will look something like this. If we expose Elodea to salt water, then it will or will not adapt. Now let's get out the microscopes and see if our hypothesis is correct. In this activity, you will be looking at the cells of the Elodea plant in freshwater and saltwater. Your teacher will make the slides and everyone will rotate through the microscope, looking first at the freshwater cells and then at the cells after they've been in salt water. To create our slides, first take your freshwater tube and you will use your forceps to remove your Elodea. You will use them to remove one leaf. The rest of the plant can stay in the tube. We'll place that leaf on a slide. You will use your pipette to collect some of the water from that same tube and just put 2 drops on your leaf. And then you will use the slide cover to put on top to make sure the water gets nice and spread out. Now you can put your slide on your microscope. While you wait for your turn at the microscope, there's another activity in your student folder for you to do. Pause the video now to start looking at the Elodea in freshwater. Once everyone has looked at the Elodea in the freshwater, you can create a slide using the saltwater Elodea. Pause the video now to look at the Elodea in saltwater. So what exactly happened when our Elodea was placed in salt water? The diagram shows that when the Elodea cells are put into salt water, water is released from the cell as it tries to dilute or make weaker the salty water surrounding the cells. This is because the water inside the cell and the water outside the cell want to be in equilibrium. What does it mean to create an equilibrium? Pause the video now to discuss. Equilibrium means that both sides are equal or balanced to each other. When the cell tried to create this balance, the cell membrane detached and shrank away from the cell wall as water left through the cell membrane. Remember, the cell wall is rigid and does not move. This forced everything in the cell to gather in the center. What do you think the leaves will look like now? Pause the video now to discuss. Since there is no longer water in the cells to keep it firm, the leaves will now look shriveled. There's no way the plant would be able to live now. This experiment shows us that Elodea is not able to adapt to salt water. Was your hypothesis correct? Well, explorers, this brings us to the end of our 3rd lesson of grasses and classes. Adaptations are super important for species to survive in their environments. Today, we learned that some organisms can adapt to changing environments while others cannot. Until next time, keep exploring.